I'm Mariangela Russo. I'm an associate professor at the University of Turin. I'm joining the group of Professor Bardelli. I'm leading on the in vitro experiment and characterization of the effect of the adaptive mutability on persister cells during their ability to evolve and to become resistant to the targeted therapies.
I'm Elisa Mariella. I'm a postdoc in Alberto Bardelli's group, and I'm a computational biologist. I'm essentially working on the analysis of all the single-cell RNA sequencing data that have been generated in the PERSIST-SEQ consortium.
The contribution of IFOM to the PERSIST-SEQ is actually mainly rely on the finding that we published a few years ago. Actually, what we found was that cancer cells, when are stressed by the exposure to the therapies, actively respond to this stress by impairing their ability to repair the DNA repair mistakes and by increasing their ability to mutagenize the DNA, with the possibility in this way that mechanism that would eventually allow them to survive to the pressure of the targeted therapies will be acquired, therefore giving them the ability to survive, avoid eradication. And this in clinical terms just turns into tumor recurrence. So based on these findings, what we are trying to do right now is to identify the key players of this mechanism of survival named adaptive mutability, because the final aim is to identify vulnerability of these cells that we can use to block the ability of these cells to survive and therefore to avoid the tumor recurrence and finally, to prolong the clinical efficacy of the targeted therapies on patients. So we are doing all these things in vitro, using different in vitro models, cell line, patient-derived 3D models, and obviously once we characterize better this phenotype, we could eventually move to in vivo characterization, finally move to patients. Adaptive mutability is a name that we gave to a stress response that we characterize in these cells. Very simply, what happens is that when the cancer cells are put in a bad microenvironment, they are stressed in their microenvironment because they are exposed to drugs that try to kill them. They activate some mechanism inside them to try to fight against this pressure, this possibility to be killed and eradicated. How do they do that? Actually, drugs induce damage in their DNA to bring them to the death. A cell line that has a damaged DNA cannot duplicate itself, but in the end will die. So they activate mechanism that would avoid this death by avoiding the recognition of the DNA damage and allow them to replicate the DNA and therefore to replicate themselves, even though their DNA is damaged. These damages are mutation. And by chance, some of these mutations can confer resistance to the effect of the drug, therefore could confer them the ability to survive despite the presence of the drug. So to replicate and give rise to the so-called tumor resistance that in patients represents the tumor recurrence. So adaptive mutability is all these mechanisms that in the end allow the cells to survive. And we're trying to figure it out how they do this to stop it. The idea is for us is always to find the vulnerability of these cell lines that would allow us to eradicate them, because clinically this cell line represents the so-called minimal residual disease. So that's minimal disease that we are not really able to eradicate from the patient. So finding vulnerability would eventually, we hope, allow us to really cure the patients.
We have a very big collection of cancer cell line that we are taking advantage for the characterization of the drug-tolerant persister cells. So we are trying to characterize different cell lines from the transcriptional point of view, and we already know that they are models in which adaptive mutability, of course. The most surprising part for me is that we technically have been able within the consortium and the efforts of all the people to find a way to obtain a very good transcriptional profile from this cell. It is, at the moment, it is not a scientific finding. It will be a scientific finding in the next future, but I think that it is really fundamental plan to build something new. So we think that this new knowledge can give us the possibility to find new methods, new strategy to counteract the onset of that mutability during drug treatment in order to delay or prevent the onset of drug resistance.
What we perform daily in our laboratory is a really translational type of research. So we always think of how to apply our finding to the patients. But let's move just one step backwards, just for a second. Sometimes we have the good question, we know what we want to study, but technically it's really difficult. So sometimes us and many other groups in the world has to face a long phase of technical setup before being able to really address our questions. So one of the huge initial steps of the PERSIST-SEQ consortium was a technical setup of performing a single-cell sequencing on this line, both from the wet biological point of view, so how to manage them, how to fix them, many of these things, as well on the bioinformatics point of view. So this first big phase of technical setup was fundamental, because without that, many of us wouldn't be able to work with these lines. This technical setup is fundamental to move a step closer to the bed of the patient, so to what is a personalized medicine.
The remaining time of the PERSIST-SEQ project, I believe that the most important point is moving from the 2D world of the cell line to the patient-derived organoids. So this is the first point. The second point may be the integration of other technologies. Some groups are starting to work with spatial transcriptomics.
So the idea is a small fraction of cells actually apparently could be really heterogeneous. So having the possibility to work with the single cell data, we could try to figure it out if really these cells are so phenotypically heterogeneous. We can try to see which of these cells are really responsible for the evolution of resistance, if this heterogeneity is maintained once they acquire resistance, whether the exposure to the drug pressure already leave a fingerprint on these cell lines. So once these cell lines are released from the drug pressure, do they keep any feature, even if they are not stressed anymore, that allow them to quickly react to a second exposure of the drug, therefore making them more resistant? So there are many, many questions that we are trying to answer by analyzing this set of data that we generated within the PERSIST-SEQ consortium.
What motivates me to work on the topic of drug resistance in general is that it's something that is really always present, despite all the efforts to find new targets and new possibility to kill the cancer cell. They always develop a drug resistance. And in particular, one point may be studying cancer resistance from a transcriptional epigenetic point of view. That is what we are also doing in the context of PERSIST-SEQ. In the last year, several new concepts are emerging, adding more and more to the genetic level.
What motivates me to work within the PERSIST-SEQ consortium is two main things: the possibility to have the access to great technologies that are actually available right now for us to really move a step forward in our research to characterize deeper what's going on in cancer cells when they are stressed with targeted therapies. But the other great thing is the possibility to be able to collaborate with the great scientists that right now are focusing on the same topic,